Good evening, everyone. We are coming to you live from the Gulf Coast Research and Education Center here in Waimama, Florida, which is on the Gulf Coast near Tampa. Welcome to Streaming Strawberries. I'm so excited to talk to you guys with some of our strawberry scientists in the Plant Diagnostics Lab about growing one of Florida's favorite fruits, strawberries. First, we're going to do a couple of introductions. My name's Bethany, and I'm going to be your host this evening. And I have 4 different plant scientists here with us today. I would like each of you to briefly introduce yourself, say your name, and what you do here at the lab.
Hi, good evening, everyone. I'm Natalia Peres. I'm a professor of plant pathology here at the University of Florida, Gulf Coast Research Education Center.
Hi, my name is Teresa Sejo, and I'm a senior biologist, and I work both in research and as a diagnostician in the plant clinic.
Hi, hello everyone. My name is Marcos Mering. I'm assistant scientist here in the GCREC Center, and I help Dr. Peres coordinating the Plant Diagnostic Clinic.
Hello everyone, my name is Andrea Marquez, and I'm a lab technician here at GCREC.
Thank you guys so much. I can't wait to ask you more questions about what you do here. But first, let's not forget, we want to say thank you and a hello to our crew. So I want to show you guys behind the scenes for a second. We have our director, Jessica, and our camera 1, Caitlin. We also have camera 2, Shannon, and our Q&A, Cesar. Say hi, guys. So don't forget that as we're doing the live stream, you are free to send in your questions. So if you have a question while we're talking about something, go ahead and send it in. And then when we take a break for Q&A, Cesar will come in and we will ask those questions to our scientists today. So don't be afraid to ask those questions. All right, we're ready to get started. We're going to start with just Dr. Natalia Peres and talk a little bit more about the center. So could you start with telling us just about the center in general, what we do here?
Okay, so we are just south of Tampa, like we mentioned before, and Hillsborough County, and that's where the majority of strawberries are produced here in Florida. About 95% of the fruit is produced around the Dover, Plant City area. If you're in Hillsborough County, you're familiar with Plant City. So the research we do here is mostly to help the growers in the area and to try to solve all the problems that they might have in their fields.
Great. So this is a really important area for strawberries in Florida. And like you said, there's a lot of growers here and they need help. But what do they need help with exactly? What are we doing for them here? Okay,
so there's a lot of different areas that they might need help with. The one that we can help in this lab and this group is mostly with diseases. There's more than that. There's often pests and other problems that they might face. And here at GCREC, we have all the different specialties. We have entomologists, we have nematologists. This group here is a plant pathologist. So we work on diseases, so we're gonna hear a little bit more later from our group and see how we actually do the diagnosis of plant diseases.
So there's lots of different roles that people can have in plant science, but here specifically you're doing pathology and diagnostics. Could you talk about what a plant pathologist does specifically?
Okay, so like I said, we plant pathologists at plant diseases. So part of what we do here is to try to identify what problem it's causing that disease on the plant, because depending on what it is, it's going to be a different recommendation for how to control. There might be cultural methods of control, maybe different cultivars that are resistant to diseases, different chemical methods, different biological methods. So the first step is figure out what the problem is. Then the next step is the research part, where we're going to try to come up with different methods to control whatever the problem is. And that's where the research part comes in. We're going to see some of that in the next segment next week. Next week or not, a few weeks from now.
Yes, a few weeks from now we will be in the strawberry fields. Yeah. But today, Plant Diagnostics Lab. Yeah. So when we talk about studying strawberries, we need to have study samples and control methods to do those studies. So how do you guys grow the strawberries for your research here?
Okay, so we pretty much grow like our growers grow in their fields. We have a research field back here where we prepare the soil just like our growers do. And in August, September, we treat the soil, we cover with plastic mulch, we put the irrigation, and then about a month later, we come with the strawberry transplants. Those strawberries are not grown from seeds. If you don't know that, they're grown from they're baby plants, like they're actually called daughter plants.
Daughter plants.
Yes.
That's adorable.
So, and those plants are grown in nurseries that are miles from Florida. They are in California, in Canada, or in North Carolina. So we plant them by hand, just like growers do. We overhead irrigate them so they can establish in this heat that we have right now. And a few weeks later, we have our plants to start our research. So we try to mimic our research fields as close as grower fields as possible.
Gotcha. So the research fields are trying to match what the growers and farmers are facing, or the growers in Florida are facing with their farms every day.
Exactly, yeah.
Great. So right now the strawberry plants are growing out in the fields. How big would you say they are?
Oh, no, we just plant them. We finished planting beginning of this week, actually. So the planting usually happens from beginning of October until about right now. So and now is the period that those plants are establishing in the fields, and that's when we start seeing the first problems coming in. Like some plants might not be growing as well as the growers would like, and that's when we start getting samples here now in the clinic so we can figure out what's causing the problems.
Could you talk a little bit about the climate of Florida and what makes it so important to understand these diseases?
Yeah, so Florida has a good weather in the winter. That's why we can grow strawberries here, but it's also warm and humid often, and those conditions are often favorable for diseases, in particular fungal diseases. That's what we mostly have here in Florida. It's a job security for a plant pathologist like us here.
Yeah, so you're going to have a job year-round because there's always going to be an opportunity for fungus to grow.
Yeah, if not on strawberries, on all the other crops that we have out there.
That's true. What about strawberries themselves? Do we grow them just one season in Florida? How does that work with the seasons?
Yeah, that's a good question. We grow strawberry as an annual crop here, and some other regions up north, they can be grown as a perennial crop, meaning more than 1 year. Here in Florida, we grow as an annual crop because our summers are just too harsh for them. So we get plants every year, usually this time of the year, October. We plant them now. They'll start producing fruit sometime in November. Usually by Thanksgiving we have some fruit, and then we'll produce until March, April, and that's the end of our season.
Wonderful. If the strawberries are really only growing in the winters, how come we see them on the shelves year-round at our grocery stores?
Yeah, good question too. It's grown in Florida that time of the year. During other parts of the year, they're grown in different areas. In particular, California. California is the number one producer. Florida is the second producer.
Okay.
But our advantage in Florida is that we are pretty much the only state that can grow during the winter.
So Florida has a unique position to be able to grow during the winter, but also that climate that really helps produce fungus, and that's where you guys come into play. You're going to study the crops to be grown in the same environments that the farmers are working with, and then once those diseases arise, help those farmers diagnose and come up with a treatment plan that's best for their crops, and that's going to make that food healthy for us too, right?
Exactly, yes.
That sounds like a hard job, but I'm
It is.
very excited to hear more about how you guys do that.
Yep.
First, we want to see if we have any questions from the audience. So this is going to be our first Q&A break. If you sent any in, we're going to try to read those. If you haven't, send them in now. So I'm going to pass the mic over to Cesar, and we'll answer some of those questions.
Hey everyone, how you all doing? Thank you for tuning in. Shout out to Volusia County, Hillsborough County, Orlando. You guys are seeing us from everywhere. That's great. But we do have a couple of questions. That someone says Dr. Chris has us showing growing vegetable gardens at home. Could we grow strawberries at home, and if so, where could we get the plants?
Yeah, so you can definitely grow them at home. They're not easy to grow, but you can. It's to get plants. We get what we call bare root transplants. Somebody can maybe grab a plant there, and I can show you how we get the plants that we grow here. Here, this is what we get. This is what our growers plant. Those are what we call bare root green top plants. You can see bare root green top, and we get millions of those in our fields. To get just a few plants for a homeowner is the hard part. There are smaller nurseries that produce plants in plugs, not necessarily like this, and you can probably find those online. There might not be exactly the same cultivars that our growers grow here, but if you can get a few plants, they're relatively easy to grow.
Here you go, David. Now you know. Stev has a question. Why do daughter's plants come from so far away?
Yeah, it's a very good question. So we, the reason in going back many years ago, I would say 30, 40 years ago, we used to have nurseries in Florida during the summer, but because of some summer heat in Florida and the rains, it was very hard, and we got a lot of disease here. So that's one of the reasons we grow them up north, where it's colder, or in drier regions like California, when it's drier, you don't have as much diseases. So those plants usually have less diseases than if we were to grow them here. The other reason to grow them up north is that for Florida, it's really important that those plants start producing early for the market. They get better price for early fruit. And the plants that come from cold regions, they come induced to start producing earlier than if we were doing that here.
Interesting. And it makes sense. And it's a great segue to this other question by Rick Hartley. It's a loaded question. Is climate change posing new challenges as far as different pests or diseases for Florida's crops? For strawberries, at least?
Yeah, it's a complex question. I climate and weather, and I work very close with a climatologist that says climate is different than weather. The weather is what we face every day. If you talk to a grower, they're often more worried about the weather, like what they're going to face this week or next week, than about climate change, what's going to happen 10 years from now. Yes, it will affect, especially like being warmer. If the warmer temperatures will potentially bring more disease problems, and strawberries often do better with cooler temperatures, especially cooler temperatures in the evenings at night.
Yeah, it was a heavy question.
It was a heavy question.
A really good question, yeah. And our last one to close up the Q&A. What made you be a pathologist, and do you like being a pathologist?
Yeah. No, I love what I do, and going back at when I was at school, maybe at the age of most of you attending, I always liked biology, and I wanted to do something that I could apply the biology to answer problems and come up with solutions. So I really find a plant pathologist being a good fit for that desire that I had as growing up.
Well, thank you so much for answering our questions, and please, guys, keep them coming, and we'll get back to you.
Thank you.
Thank you, Cesar. Thank you, Natalia. I really loved that last answer to the question about why you do what you do, because this is an IFAS facility, so Institute of Food and Agricultural Sciences, and that means that there's tons of research happening here every day to make life better for the people in our community, and in this case, it's for those strawberry farmers and growers. So it's really cool to see that play out live. Speaking of which, we're going to talk about Teresa's work here now, diagnosing. So we use the scientific process all the time in our daily lives, but we don't always think about it in specific details. But if we lay it out, we see that happening right here in this Plant Diagnostics Lab. So the first step is observation, right?
Yes, and we actually start not with our own observation, but with the grower's observations. So if the grower is having problems, if they notice something's not right with their plant, they're going to bring us samples of those plants, and we're going to ask them to fill out a form for us. And in that form, we're going to ask a lot of questions, and the more details they can give us, the more it can help us diagnose, give us a clue of what's going on. Things like knowing what cultivar is being grown, because different cultivars have different susceptibilities to different diseases, how long ago it was planted, because the age of the plant can affect the susceptibility, what the local what symptoms they're seeing in the field. Is it all their plants? Is it in just a limited area of the field, like maybe like a very low area of the field that might be prone to flooding, or just one particular bed might have a disease issue? Sometimes those patterns can help us determine what's going on. We also have to consider the weather patterns, because that can affect if we've had a lot of rain or if it's been excessively hot, that can affect what we're seeing in the field.
So the first step is that the farmers make an observation. They have a concern about some of their plants. They bring those plants to you. Now you need to make your own observations as well, right? So you're comparing that to the information they gave you?
Yes. So the
Okay.
first thing we're going to do is just take a look at the plant and see what we can see.
Let's take a look.
And so these are some plants that I actually pulled from our field today. And these ones were nice, healthy plants. I just want to give an example of nice, healthy plants. And these are plants that were not establishing. They're wilted. They barely grew. You can look at the difference, not just the size of the plant, but the root systems. The difference in the root systems and the amount of growth. All these plants were planted about 2 weeks ago. They're from the same beds, the same plants from the same beds, and you can see a vast difference between these plants.
So sometimes the plant is facing an issue that's not always a disease, right? So what are some other things that might be causing a plant to look
Yes.
sick like that?
So sometimes we get issues that are what we call abiotic problems, and these can be things like an excess of a fertilizer causes what we call high salts, and you can see actual burning, the leaves at the edges of the leaves turning brown. You can see if it's really high, it can actually cause the plants to die. It can affect the base of the plants and cause the plants to start wilting and dying like that if it's very, very high. If you don't have enough fertilizer, you can start getting nutrient deficiencies, and you might get a plant turning yellow, or you might get some red hints on the leaf that indicate various nutrient deficiencies. If something has happened, like say the irrigation system got clogged, the plants may just not be getting enough water, and sometimes that can affect.
Right. So there's so many things that could be happening. But let's say that you do end up forming a hypothesis that you do believe it is a disease. What are your next steps for testing that hypothesis?
So what we're going to want to do, so diseases caused by biotic problems or living organisms can be caused by a number of different types of pathogen groups. For example, we can have diseases caused by viruses, by fungi, by bacteria. You can also get problems caused by little microscopic worms called nematodes, and of course insects. So part of it's just observation. So are we seeing leaf spots? Are we seeing the plants wilting and dying? How do the roots look? I might get it and actually look for the physical presence of insects, or do I see evidence of feeding on the plants? Something was actually chewing on it, eating it. So start looking at those things.
Is this a good example of some leaf spots that we could look at too?
Yes, that is. So we have some.
So you're still going to make some observations before you start testing anything?
Yes.
Okay.
Based on what we're seeing, if I think it is an insect or a nematology problem, I might bring in some experts in that area. I might take them some samples. I might say, okay, I'm pretty sure this is a nematode problem, but they would be better suited to identify which nematode is causing the issue. And we are fortunate that we have nematologists at the facility that help us out on those things.
Very convenient.
Yes. So we deal a lot with fungal and bacterial diseases, which strawberries, we're fortunate, usually don't have a lot of problems with viruses. They can, but they don't usually. So we do a lot of work with fungi and bacteria, and on strawberries it's mostly fungal problems. So this is a perfect example here. I am going to take a second here and cut open a healthy plant and a diseased plant here, and I want to show you what our crown looks like. And the crown is the base of the stem, the part that's right in the ground, that's at the soil line where the roots are coming out of. We call that the crown of the plant.
That sounds great. We can't wait to see it. So first, Teresa is coming over here and she's going to cut into the healthy plant with her scalpel very carefully.
So this one's interesting because it is mostly healthy, but you can actually see a little bit of discoloration here. That could actually be early disease symptoms forming that we can't see any symptoms on the outside of the plant yet.
So to be continued.
But generally, a healthy crown is going to be pure white with no brown inside of it.
Should we cut open another one?
We can try, yeah.
Let's see what we got. Plant number 2. She's using that scalpel to cut open that crown. Let's see what's inside. Looks like kind of another yellow one.
It does have a little bit, but this is actually just from experience. I know these little tiny brown flecks are actually usually a stress symptom. So that could just be transplant stress.
Gotcha.
Sometimes it gets a little...
These are daughter plants too, right? So they've been recently
Yeah,
transplanted.
these were planted 2 weeks ago, and I just pulled them out of the ground. So they just... Let me try...
Well, let's compare to a plant that we can tell is looking much worse than our healthy plants and see what's inside.
Well, as opposed to that little bit of brown, this one is completely, the entire crown, inside of the crown is brown now here. We call this a crown rot. And
All right. Should we try one more?
I'm going to try one more.
You guys are getting a lot of really good samples here. And this is also a great example of science in that sometimes it's not always what you expect it to be, right? So we thought we might see a plant that has some fungal growth and some not.
And here,
But
this is what I was
we'll
hoping
see.
to see, where you have this whole bottom area is all crown rot, and here you see the top of the plant that's still healthy. Still looks like a healthy white crown.
Beautiful. So we have our sample and we're ready to look at it under the microscope.
So this is, what I do then is that there's a number of different pathogens that can cause this. So looking at this, I say, okay, I know this plant has got crown rot. This is killing the plant, but now I got to figure out what's causing it. And there are a number of different fungi and one oomycete that can be causing these symptoms. So we're going to plate it out and we're going to see what we can actually isolate from this tissue. And
that's why it's called traditional isolation.
yes.
Yes. Traditional isolation. So we're going to get an example of that now. But first, Teresa needs to sterilize her equipment. So she's going to use her lighter and some rubbing alcohol and sterilize her scalpel with the flame and the alcohol.
Okay.
And she's going to get the microscope prepped so that we can get a good view of what we're seeing under there. Takes a little bit of dialing in. Once again, science in action. Good things take time.
Okay, that one. Okay.
So re-sterilizing her scalpel real quick.
I set it down so it's no longer sterile. So I am actually, and I'm going to flip this around. I'm flipping this around because I don't want to be cutting towards myself. So I am going to
Basic scalpel safety.
cut away, and I'm just cutting a thin layer off with the sterile scalpel so that I basically want to make sure this is clean and that we're only getting from the inside of the plant.
And then doing one more round of sterilization.
And when I isolate, I want to isolate right on the edge from this brown necrotic crown rot tissue where the pathogen will be moving into this healthy tissue here. So I'll cut samples right from the edge there.
All right, I'm gonna give her a little bit of space while she makes this very small incision. And like she said, she's looking for that line of where the healthy tissue is meeting the decaying tissue. And she's just gonna take a really small sample. You guys are gonna see how tiny it is on the scalpel. So very precise work. Let's see if we can see it with our cameras.
And then I take this and I'm going to place it on a petri dish. You can see it up there. You can see the dark and the light side as I put it on there. And then
So that's just one sample from one plant.
So if when I'm doing this for real though, you can't just do one sample. So I'll take multiple plants and take pieces from all of them. We want to find out what's going on in the field as a whole, not just one particular plant that's already not in the field anymore. So I want to know what's going on the field as a whole. And the reason why we get that right on the borderline is because that once the tissue starts dying and turning brown, we can get saprophytes or fungi that aren't really pathogens but just come in and like to feed off a dead tissue. So you get bacteria, fungi, worms, all kinds of things that aren't pathogens that can't directly hurt a healthy plant, but they can get into that dead brown tissue.
So those,
just the things that normal decayers.
yeah, so those unwelcome visitors are going to be making it harder to diagnose.
Yes.
And what do you do with the sample from there?
So from here, so this is just, the Petri dish is basically just food for the fungi. So this is actually made of, is potato dextrose, so it's potato and sugar. Hard to grow on.
Yum.
And so a little bit of antibiotics to prevent bacterial contamination. And we'll grow this for about 5 to 7 days, and then we'll see what grows.
I heard a rumor that you guys also feed them V8 juice. Is that true?
We do. We do. Some of the fungi really like V8 juice.
Is there a special flavor they like?
We get just traditional.
Traditional. You heard it here
Regular,
first.
not low sodium, just the basic traditional V8.
Traditional V8 juice, folks. All right. Thank you so much for showing us a piece of your work. I know there's more to be said about the fungi and all of the Petri dishes. We're going to transition to questions, but bring your Petri dishes with us and we can show some close-ups of what that looks like as they take time to grow.
Okay.
Cesar, passing it over to you.
Just wait for. Oh. All right, that was a lot of movement, but I'm back. Hello, everyone. Thank you again for your questions. We're gonna get to as many as we can, but let's just get right to it. Okay, so Sean, it just moved. Okay, Sean Busby says, what disease or biotic factor is most common in your experience?
It varies from year to year. So we have some diseases that we always see almost every single year. Actually, there's quite a few, but for example, Phytophthora crown rot is usually an establishment problem. The plants come infected, and we get a lot of, when we establish the plants, we put 10 to 14 days of overhead water, and that pathogen really likes water. So we get a lot of early symptoms. We almost always have some level, some botrytis fruit rot, anthracnose fruit rot. But many years it'll be at low levels, but then you'll get a year where it'll peak and you might have, I've had years where we had like 80% fruit loss, some growers did. So a disease that might normally be present at low levels of like 5% or something, and then you can have peak seasons when the weather's right and you get a susceptible cultivar. And there's a number of diseases that can do that under the right conditions. Yeah,
it makes sense. It varies, I guess. And is it, are those usually bacteria or viruses the most common?
In strawberries, fungal diseases are the most common problems. We have 2 bacterial diseases, but they just cause leaf spots, so they don't cause plant death and they don't really cause fruit rot. So they can decrease fruit yield, but they don't cause this extensive problems.
I see. Okay. Sophia, another loaded question, so get ready. Sophia says, do you treat pests such as nematodes organically? Do you use IPM? What is your definition of organic treatment?
Ooh. Well, organic treatment is a legal definition from the government, and they have very set definitions of which chemicals qualify as organic and which chemicals are not organic. So if something is a lab-produced chemical, it would not be organic. If it's a naturally derived, if it's coming occurring more in nature, not being produced in a lab, then it's could be considered organic, but it's still a chemical treatment. And that's important, an important distinction that people do need to know. It doesn't mean it's completely chemical free if it's organic. But strawberries, most strawberry production in Florida is conventional. It is not organic. There is a little bit of organic production in Florida, but it's challenging because our conditions are so conducive to diseases here. So you're best off, it's easiest to grow organically when you have low disease pressure. When you have high disease pressure, it can be very challenging.
Makes total sense, and it seems like Sophia knows a bit about pathology.
We do test organic products, and there are some common what would be considered organic products that we use on a regular basis when they're effective. We do love to use predator mites in our fields to where we actually release little predator mites that will eat the mites that damage the strawberries. Sulfur, which can be applied for control of powdery mildew, is quite effective, and it is considered an organic chemical.
Yeah.
So we do use some organic chemicals in our product.
Yeah,
if they're effective, we use them.
That's great to know. Yeah. Another question: can you save a farmer's crop if you identify the fungus in time from your diagnostics work?
Sometimes we hope to. That is our goal. Sometimes you don't save 100% of the crop, but you save a certain percentage. So you decrease. If they get to us soon, they might have 20. They might be at 20% loss, and by getting the right treatment on it, we can keep that loss low instead of having it escalate to a 50% or a 60% loss. We can keep it down at 25% or something.
Yeah.
So we might not completely control the disease, but we might be able to keep the losses, decrease the losses they'll see.
Understandable. Hopefully enough for them to bounce back. Thank you so much for answering our questions. We'll keep bringing them back when we get them, but let's get back to you, Bethany, and let's keep talking.
Thank you, Cesar. That last question was another perfect segue to our third and final segment, because we were talking about how important it is to have a timely diagnosis. So with the methods that Teresa was just showing us, those traditional methods, we're depending on the morphology of the petri dishes, or what we're looking at in the petri dishes, right? So we have to look at it by eye, try to identify from there. But Marcus, could you tell us how long those traditional methods take?
Yeah, so usually like the traditional isolation or the traditional methods that we look at morphology and how the pathogen looks like on those culture media, on those petri dishes, it can take up to a week or even more, depending, because some pathogens, they have a slow growth pace on a culture media, so it depends on the case, but usually on average 5 to 7 days we should have an answer, okay? So here in our table, we have some of the strawberry pathogens grow on the medias that we use in our clinic, and that's very nice for you to see how they look so different from each other. It's like a different person, a human being, you know. Some of them produce a pigmentation on the media, so maybe the media change of the color. So it's very interesting to see, but like we were mentioning, it takes time, and to save a farmer and to save the plants that are sick out in the field, we have to have a faster answer than the traditional methods.
Right, because 5 to 7 days doesn't sound like that long of a time, but when you think about how fast these fungi are growing just on these petri dishes, you can really imagine how fast they're going to take over those farmers' plants. So could you tell us what you're doing to help that? What are you doing that can make that process faster?
Sure. So here in our diagnostic lab, there is a commercial kit that is like a COVID rapid test. Okay, so we get a symptomatic tissue, and then we smash it into a buffer or a solution that comes from a company, and then if we have 2 lines like it is on this test, it means it's positive for the pathogen we are testing. In this case, the pathogens call it Phytophthora. Okay, so this is a very a simple analogy of like, okay, the plant diagnostic clinic is like a walk-in clinic that you go and figure out what disease it's going on, not with humans, but with plants.
Right.
Okay.
So this rapid test is a great way to get a fast result. However, it's just for one species, right?
Mm-hmm.
So what do you do if there's a different species and it tests negative in the rapid test, but you still need that fast result?
So if it is a different type of pathogen, we have here in our lab, we develop some PCR tests. So PCR is like a reaction that is based on the DNA that we extract from the symptomatic tissue. And here we have lucky because we have this machine that was donated by the Florida Strawberry Growers Association because they really appreciate the work we do, so they donated the machine to us. So on this machine, what we do is like Andrea is holding here as an example. We have a special plate. On each well, we can put a different sample to be tested. So on that well, we put DNA samples from the samples that the growers brought in. And then we put the plate inside of the machine. We mix some ingredients to do that reaction. And basically what this machine will do is going to read the DNA that you put it on each well from each sample. And like us humans, we have our fingerprints that we can recognize how one person is different from the other. So this machine is going to create what we call it a different DNA curves, is the way that the machine presents the result to us. So different curves, it means different DNA of a specific pathogen, okay? So in this case here, for example, you have a blue curve, a red curve, a yellow, and a pink curve. Each of these curves is the different pathogen, okay? So once we put at the sample, for example, this sample here is the ones that are labeled in green, okay? So green, it means it reacted, okay? So in this case, since all the greens match it, the blue curves, it means we have that blue pathogen. In our case, we are coding that blue pathogen has Phytophthora. Okay, so in that case, the sample is positive for Phytophthora. And this result, we can give an answer to a grower within 24 hours after the sample submission. So they can be ready to go to take an action to save their plants in the field.
Wow, so way faster than 5 to 7 days. It just takes 1 day. And whereas the traditional methods are looking at morphology or what's on the outside, this is testing DNA of the pathogen, right? And the pathogen, could you explain in the disease triangle, what's the pathogen versus the disease versus the host and the plant?
Okay, sure. So what happened is like the pathogen is the microorganism that is causing the disease.
Okay.
Okay. The host, in this case, will be a strawberry plant that is susceptible to a pathogen. And the other part of the triangle, you have the environment. Okay, so whenever all the three sections of the triangle react together, you have the disease. So actually the disease is the host interacting with the pathogen and also interacting with the weather. If you have one of the missing pieces, you don't have disease.
Great.
Okay.
So when disease is present, it's so important to make sure we identify that pathogen. And this is allowing us to identify that each pathogen has a unique curve. And when the sample is reacting with that curve, we know it's that pathogen.
Yes.
Wow, really incredible technology.
Yeah, and one of the nice things about this technology is like all these pathogens we are testing, they are mostly fungi. But it's not because they are fungi that you apply any fungicide that you control them. Each microorganism have a specific management recommendation or a different product that needs to be applied. Like us, okay, maybe I have a sore throat or I have a different disease. The doctor will recommend you a different medication. So it's the same thing for the plant. So it's really important to know, not really that, okay, this is a fungi, but what type of fungi? What microorganism are you having there?
Right, because you don't want to over-treat the plants. You want to give them just the right dose of just the right medicine
Yes.
in a timely manner.
In a timely manner.
Awesome.
And that's very interesting because one of the things that we are working here in the lab too is to develop an app for the cell phones that actually growers or whoever is out in the field, they could take pictures, for example, of a leaf spot, and then if they submit this, for right now it's like a prototype of the app, so it's not like a open access yet. But here is just an example. It's like you take a picture of your leaf spot symptoms, and then you submit it to the app, and the app will give you like a score or similarity to any of the disease that we are loading and feeding that app.
Wow.
Okay, we started doing that because we had an emerging pathogen on the strawberry that was not here before. It's just started recently. That's caused by a fungus called Neopistolochiopsis. And this disease was like the COVID for the strawberries, you know. It just emerged, it started to spread throughout the field, and we didn't know how to control. We had to do some research to figure that out, and it was really scary for the growers, so we had to take fast actions, like developing the DNA tests, developing an app that the growers could detect that disease, so you can really help them figure out what's going on with their plants.
Right. So just as new diseases are evolving and emerging, so is the technology that you guys are using to help those farmers address those issues. That's really incredible. Thank you so much for sharing all of that
You're
with
very
us.
welcome. Thank you.
I think we're ready for our final Q&A. So if you didn't get any questions in last time, now's the time to send them to Cesar, and we'll hopefully get them answered right now for you.
Thanks again, Bethany, and hello, everyone. We have more general questions, more general plant disease questions. So
Okay, no problem.
Let's see, and just plant questions. This one from Alatra EDP. How do plants get stressed?
Okay, thank you for your question. Yes, like we were talking before about the disease triangle, like the host, the microorganism, and the weather, the plant gets stressed for any of those different points of the triangle. It could be a heat because the environment, or it could be too much water, or maybe it could be a human activity like the excess of fertilizer, or maybe the cultivar or the variety that you are using in a certain area is not adapted to grow in that condition, you know. So all the ways that I think like a human got stressed, a plant got stressed too.
Yeah,
Okay.
it does sound that way, and Florida seems to be stressful for
yes.
strawberries. Yeah, and for us too. Hannah Kim says, what is the downside of DNA testing?
Okay, so the downside of the DNA testing would be, in some cases, for example, if the grower applied a fungicide and they send us the sample after the fungicide application, it might be positive on the DNA test because the DNA is still in there. But whenever you do the culture isolation, it might turn out that the fungicide already killed that pathogen, and therefore the grower doesn't need to do any extra action because what he have done, it's already solved the problem. Okay, so that could be one of the downside of the DNA. It might be positive for a disease, you report to a grower, and it might happen that he already have take care of the problem. Okay.
It could be redundant at some point.
Mm-hmm.
Yeah,
Yes.
understandable. This one from Hannah Kim. Again, do you think coding or computer science is becoming a big part of the agricultural industry?
Yes, sure. That it's like it's already merging into our program, our science, and even like in our research center. Probably you have heard about it, the AI center that they are building here next to us. And that's an example of the app that we are developing to diagnose the diseases, you know, is just bringing in the computer science training models and translating those to daily routine that growers could use to save their farms and continue their production system.
So it seems like coding and computer science is also another option if you want to get into the agricultural
yes.
industry. Yeah, great. Technology is going to help us do things better for sure.
No, for sure.
And this one to close up, and it's a fun question, so you get to answer a fun question. Alachua EDEP says, this is a general question from a 7th grader.
Okay.
Which is great. I heard that there are strawberries that taste like pineapples. I heard that they're called pine berries. Is that true?
Yes.
I want to know too.
That's actually true. So we, Dr. Peres collaborate a lot with the breeding program here in our research center, and they develop a cultivar of strawberry that produce white berries. Okay, so those white berries, they really taste like pineapple. They have a different color. They are very tasteful. So if you haven't tried yet, I think you should. And I'm pretty sure you're gonna love it. They're very different and they're very good.
So they look like a strawberry, but they're white, or they're different colors?
Yes, they looks like a red strawberry, you know, but they're just white.
Okay.
Yeah.
So their color's white, and they just taste like
like pineapple.
The pineapple, or whatever they decided to do. And is there a place where we can buy these?
Oh yes, like starting like last year, they already start selling at Publix and some grocery stores, so you can easily find them nowadays, especially now that our season started. So keep on the lookout next month.
Amazing! Wow.
Okay.
Well, let's go get those strawberries too. And thank you so much for
You're very
answering
welcome.
all our questions. And thank you guys for asking the questions. And I'm signing out for now.
Thank you, Cesar. All right, guys. Remember, we're going to be back here in November, and we're going to be in the strawberry field, so you're going to get to see those strawberries growing. And I just wanted to say thank you to the team here at the Gulf Coast Research and Education Center, the Plant Diagnostic Lab team. Let's all get together for one final thank you, and thank you to everyone for watching today, and our whole Streaming Science crew for being here to put this all together. One final note. Thank you to everyone who helps make Florida's strawberry industry a little bit sweeter. Well, just cheers. Oh wait, Andrea needs a strawberry. Yay!
I heard it's her birthday today, so we're gonna cheer for her birthday.
Yes.
Yeah.
Thank you guys so much.
birthday.
Streaming birthdays. Thank you. It doesn't taste like pineapples.